And I said a couple hours. Okay. Online, can you hear me?
Yes.
Great. I'm gonna turn on the projectors. Awesome, thank you. So we're gonna pick up where we left off. Yeah, you can see my screen. Okay, great. I'm just waiting for our projectors to go. So when we left off before the break, we had gone through glycolysis and we've converted pyruvate to acetyl-CoA. We're gonna now talk about the other fate of pyruvate, which is an anaerobic glycolysis and where that goes, which is really, it becomes lactate. It's not super exciting. And then we're gonna enter into the world of the Krebs cycle. At the end, I have some kind of summative images, pictures, things at the last slide. It's towards the end of the last slide set. I'll show you them. We probably won't like review them in class, but they're really great pictures for you to practice putting all these pathways together. And so I encourage you to utilize them in your studying, cover things up with like Post-it notes or little pieces of paper, you know, quiz yourself, check your answer. It's a really great way to do some high yield study for more of those longer processes. Okay, great. We're here. Yes. All right. Okay. So looking at the fate of pyruvate, so anaerobic glycolysis, this is really our anaerobic respiration. So if we do not have oxygen present, this is also occurring in the cytosol. It is another kind of side benefit. It can help us eventually be able to regenerate NAD+, which we can then use in glycolysis to generate NADH. So this can also be an added benefit for us if we end up undergoing glycolysis further, if we are intaking more glucose. And it will occur normally in cells without mitochondria. So this is your metabolic process of your erythrocytes, your red blood cells. So great NPLEX question, what cell type undergoes anaerobic glycolysis under oxygenated states? It will be your erythrocytes or red blood cells. They don't have mitochondria, so that's why they cannot undergo our typical glycolysis to the Krebs cycle because there's no mitochondria to go to. In skeletal muscle or other tissues, this will occur during anaerobic conditions, and then from there, we will have to deal with the lactate buildup, and so we'll show kind of some processes later on where lactate kind of fits in and how we can convert it back to something more useful. It does cost energy to take lactate and to convert it into a useful substance that can be used in the body again. So this is an energy depleting process at the end of the day. It gives us a little bit of energy by going through glycolysis, but because we don't get that NADH, because lactate doesn't go through the Krebs cycle, it is not going to be as energy rich as our typical pyruvate Krebs cycle phosphorylated oxidation. Okay. Moving into the TCA cycle, the Krebs, you're going to be happy because we're not going to spend a lot of time here because there's not a lot you have to know. So TCA, Krebs, citric acid cycle, all same. So what are some of the sources of acetyl-CoA? We've talked about pyruvate as being one of the main sources. Amino acids, alanine, as well as glutamine can also be feeding into this process, and they can be converted into pyruvate. Ethanol can go down this process. Sugars obviously get converted into pyruvate. Ketone bodies, acetoacetate can become acetyl-CoA, and fatty acids, this is an example of palmitate, can become acetyl-CoA. So there's lots of different sources. We don't just have to think glucose here. So anytime you have acetyl-CoA, regardless of the source, we can go through the Krebs cycle. And this is kind of just showing the relationship between ethanol, acetaldehyde, and acetic acid. One moment about amino acids. So I mentioned that amino acids can be utilized to create acetyl-CoA. There's two kind of types of amino acids, ketogenic versus glucogenic amino acids. And we'll look at ketogenic amino acids and ketosis and that process a little bit more later on. It's in my last section of our review today. But ketogenic amino acids can be oxidized to form acetyl-CoA for ketone body genesis during ketosis. The only two ketogenic amino acids are lysine and leucine. So that's why it's a good test question because there's only two of them, right? So lysine, leucine are your two ketogenic amino acids versus PIT, phenylalanine, isoleucine, tyrosine, tryptophan, and threonine can do both. So that's 5. So that's pretty unique as well. So they can do either ketogenic amino acids or glucogenic amino acids. All other 13 amino acids are only glucogenic, meaning that they're converted into glucose for gluconeogenesis. So our ketogenic amino acids can go to become ketone bodies. Our glucogenic amino acids can become glucose. And then our the PIT, which is our mnemonic for those 5, can do both. They can do either ketogenic or glucogenic amino acid conversions. You can kind of see where and how some amino acids are going to be converted into different parts or different areas. So lysine and leucine, acetyl-CoA, and then we see kind of these ones in purple. These are both ketogenic and glucogenic. They can become acetyl-CoA, they can become pyruvate, succinyl-CoA, or fumarate. And these are components of the citric acid cycle. The ones in red can only, they're only glucagonic. So aspartate and asparagine can become oxaloacetate, alanine, glycine, cysteine, serine, pyruvate. We have these 4 that can become glutamate, which then can become alpha-ketoglutarate. These 2 can pop in here. So you can kind of get an idea of where they can come in. I would not memorize this chart outside of knowing that lysine and leucine are the unique ones that can become ketones. All right. So the citric acid cycle, the TCA cycle, occurs in the mitochondrial matrix. So we've moved from the cytosol to the mitochondrial matrix. So acetyl-CoA is able to move, right? Pyruvate is able to move into the matrix, the mitochondria, and then acetyl-CoA is present there, and so it's able to be utilized in this cycle or amongst other things. The rate limiting step, what's our rate limiting step of the citric acid cycle? Yep, isocitrate dehydrogenase. A close second, you could say this is also the rate limiting step, is going to be alpha-ketoglutarate dehydrogenase. So these 2 are going to be your rate limiting steps. The reason I chose isocitrate is it comes first, but both can be rate limiting and both will be rate limiting. They're dehydrogenase. So what type of reactions are they governing? They're a dehydrogenase enzyme. There's NAD involved. So what type of reaction must they govern? A redox reaction. So these are both redox reactions. And we're going to have another one later on, succinate dehydrogenase and malate dehydrogenase. All 4 of these enzymes are all going to govern a redox reaction. So these are all important reactions because isocitrate dehydrogenase comes first and alpha-ketoglutarate dehydrogenase is also an important enzyme, comes next. These 2 are going to be your rate limiters. Okay. From here, the only other pieces I would really lean into, no, obviously acetyl-CoA plus oxaloacetate, OAA sometimes or OXA sometimes. These 2 are going to be your starter molecules of this cycle. So acetyl-CoA, oxaloacetate are then going to combine together to form citrate. Citrate, yes, related to citric acid. So that's where your vitamin C connection comes into play. We then see that aconitase, that was a test question, I believe, earlier. I forget what it was about. We talked about it. And then from here, we have our 2 rate limiters. During our 2 rate limiters, that's where we get 2 of our NADHs. We also are removing 2 carbons, right? Because we start with a 2-carbon molecule and a 4-carbon molecule. And the first step of the TCA cycle is we combine them together to get 6 carbons in citrate. We then are going to cleave off a carbon here, cleave off a carbon here. Then we stick with 4 carbons for a while till we get back to the beginning. These carbon dioxide molecules end where? Where do they end up going eventually? Not necessarily, although they can be used. This process as a whole, from glycolysis all the way to the electron transport chain, what do we call it? This whole big process using oxygen. That's the end part, oxidative phosphorylation, yes. From start to finish, it's called some type of something. Aerobic respiration, right? Aerobic respiration. So in aerobic respiration, we have to have oxygen to do this process. And our byproduct is carbon dioxide. So physiologically, this carbon dioxide is probably going to go diffuse to where and end up going where? The blood, and end up going to the lungs to exhale in expiration. So that's your physiologic connection, right? All these cells in our body are doing aerobic respiration as long as we have fuel to do so and the oxygen to do so. So we all just ate, and we all probably ate some form of carbohydrates, whether it's a vegetable or straight on starch. Some of us probably had some fructose, sucrose, who knows what, right? And now our bodies are breaking that down in our digestive tract. And as they get broken down into monosaccharides, they're undergoing this literal process here for me to continue to talk to you, right? That's what's happening. And as we're exhaling this carbon dioxide that we're creating as a byproduct going through the Krebs. So I want you to like keep in mind like the big picture, even though we're super small in the weeds molecularly, because it's helpful. Okay, so we go through this whole process here. We get an FADH and we get another NADH. So if you wanted to memorize more than just isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase and its substrates and its products, you could focus on the other two dehydrogenase reactions. If you wanted to go crazy, you could do the whole thing, but at the minimum, do these two reactions. So isocitrate to alpha-ketoglutarate to succinyl-CoA. That's your minimum for the Krebs. If you can know more, go from succinate to fumarate and then malate to oxaloacetate. If you can know more, sure, memorizing more, knowing more is always going to be better. Okay, but I'm trying to give you some scale. So if you don't want to have to learn the whole thing, you could try not to. That's it. Krebs cycle. Done. That's all I want you to know. Yeah. Hey, the question was, what's the purpose of the TCA cycle? So I'm going to ask it back to you. What is your purpose in your mind? And then I'll go where that question is going. Uh-huh. True. Which direction? The main one. Okay. So we think about the TCA cycle in school. What is it really doing a good job at getting us? What do we get a lot of at the TCA cycle as a product? NADH, right? Our products, if we think about this, for the most part, unless we have a deficiency elsewhere, these like substrates and products are staying here in the mitochondria. They're not really leaving. They're not really going anywhere else. Sometimes we'll pull some to go elsewhere. Sometimes we'll put other things in to replace them if needed. But they're mainly just like kind of hanging out. They don't hang out in a circle, but they're hanging out in the mitochondrial matrix. Our products are all these things in blue, are actual things that we're getting. So all of these NADH and FADHs and the CO2s, but they aren't really a helpful product. All of these are the products. So I'd say the purpose would be to harvest those NADH, FADH molecules, which what are they holding on to? Those NADH and FADH molecules. What have they taken in this process? Yeah, so we're electron harvesting. So if I had one purpose of the TCA cycle, it's electron harvesting to eventually make energy in the form of ATP. So you could say energy generation, you could say electron harvesting or electron gathering or electron generation. I think any of those would be probably the most appropriate answer. And I think there was one answer that said like electron something, and that would be the answer I'd look for. Yeah, excellent. Good question. So speaking of electrons, so once we have these molecules, now we're going to move them through the electron transport chain, which is in the innermost portion of the mitochondrial matrix. And so we have 4 or 5 complexes we want to pay attention to. So complex 1, 2, 3, 4, and 5. What I really want you to care about is not necessarily knowing the exact names or all the different things that can affect each complex. But we're going to pay attention to a couple components, a couple like prosthetic groups that are important, and cofactors that are helpful to know. There's lots of cofactors used in the TCA cycle, absolutely, Gigi. So we're going to circle back to cofactors in the TCA cycle in a little bit. But yes, there are lots of cofactors in TCA. Pretty much anytime you're redox reactioning, there's cofactors, right? All of your NADs and your FADs are considered cofactors. So you can, at the minimum, consider those. And then there's several different enzymes that actually mimic pyruvate dehydrogenase complex that have that list of 5 cofactors again. So there's a lot of cofactors that do come into play. Oftentimes we're asked the most about NAD, FAD when it comes to the TCA cycle. So in our ETC, so complex 1, we start with our NADH. So that's the first complex. That's the first thing we're going to harvest off. We're going to harvest some hydrogen ions, which is an electron, right? So think of protons as electrons in this, and I know it's weird, Okay, so I want you to think of protons. We're creating this proton gradient by taking electrons off of the molecule. Okay, so we have NADH. We're gonna take away the electron. So if we're taking away electrons, we're doing what to it? Yes. So we're taking away electrons. And then from there, and those electrons, that's helping us move the things from, the electrons are moving, they're helping us move stuff from one thing to another. What we're moving is we're creating this proton gradient. That's what all these hydrogen ions are here for. So H, 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 and H. This proton gradient is going to create a charge across our membrane. Anytime we create a charge across the membrane, it's going to cause some type of action to happen, right? When we think about depolarization and repolarization and action potential, it's because we've created this electrochemical gradient across of our membrane. And so we can have things move across it. That's what we're creating here in the electron transport chain. It's just with hydrogen ions. So we get this big plus positive charge in this intramembranous space. And we're going to use this plus positive charge to actually help us create ATP using this protein that literally is like a stick with a rod on top, and it twists. And when it twists all the way around, it pops out a molecule of ATP. And it's from these hydrogen ions actually triggering and binding and creating this space, this electrochemical gradient. These hydrogen ions go in here, and they're able to actually help us turn this molecule around and give us ATP in the form of energy. So that's what we're doing here, is we're harvesting electrons in this reaction and in this reaction. We're passing them along from one complex to the next, to the next, to the next. And with doing that, we're harvesting hydrogen ions and creating an electrochemical gradient. And so that's the whole process here, right? The phosphorylation comes into play because that's the goal, right? We're trying to take a molecule of ADP to phosphorylate it to give us ATP at the end. And that's the end all be all. That's what we want to do at the end of this process, okay? Now, where it comes important is to pay attention to a few of the cofactors. So this is where I see the most common test questions for NPLEX. So one of the test questions is, where does CoQ10 come into play? So we have this little CoQ10 molecule. So we'll look at here, this next page. So our first enzyme complex, number 1, we're dealing with NADH, and this is a flavin mononucleotide. So we're dealing with riboflavin, so B2 is going to be a cofactor. And we're dealing with a sulfur and an iron. So iron is also a cofactor here. So patients who have iron deficiency, don't have adequate iron stores, they could potentially see effects in some of their electron transport chain processes. Although the body typically will use the iron first here versus other things. Succinate-CoA reductase, we have another FAD. So this is another riboflavin B2 molecule, and we have another iron cofactor. Our CoQ, or our cytochrome C reductase, this is where you have your CoQ10 comes into play, and iron again. These are just other molecules of heme. Heme binds to iron, so it makes sense that these are here as prosthetic groups. Cytochrome C, I don't have any fun prosthetic groups. And then cytochrome C oxidase, we have copper. We haven't seen copper before, so this is a unique one. So we pass our electrons down the chain and we create a proton gradient, okay? So this is the mitochondrial matrix. This is the intramembrane space. As we are kind of entering into here, we're going to track things from start to finish. So ADP is moving inside of the mitochondrial space, as is pyruvate. We then undergo the Krebs cycle, all those good things. And then ADP will actually move inward into the inner mitochondrial matrix in exchange for ATP. So this is an antiporter. That's what ANT means. Within the mitochondrial matrix, we're passing along our molecules, our NADH molecules, from one thing to the next, and we're creating this proton gradient. So here's all of our little happy protons. This is the electrochemical gradient that we see here, this big positive charge, and then it's negative on the inside compared to the outside. Because of this positive gradient, that is what's going to trigger this. I don't know why it's doing little weird, crazy things. This protein complex, this is your ATPase, to phosphorylate ADP and create your ATP. The ATP is going to bump into this innermost mitochondrial matrix. Here. How do we get the phosphate? There is a phosphate hydrogen ion symport. There's plenty of hydrogen ions outside. So this hydrogen ion complex is going to drive phosphate inwards because there's more hydrogen outside than inside because of this proton gradient. It's also going to drive in these 3 carbon molecules. We have another symport here. and it's also going to drive in calcium. So this is what it looks like from an actual biochemical perspective. From there, once the ATP is created, it's going to, again, antiport out with ADP coming in. And then we have our ATP molecules here, which can be utilized wherever is needed, wherever is necessary. So now is the big question. How do we generate energy? So there's NPLEX rules and there's reality. So I'm going to teach you both because I want you to know both, and NPLEX could ask you either, but historically NPLEX has used NPLEX rules. And I say this because NPLEX is typically like in the olden times, sometimes. So traditionally, NPLEX will say that 1 molecule of NADH will equal 3 molecules of ATP, okay? In reality, it's 2.5. So if we're being perfect, it's 2.5, but we're going to say 3 for now because we're doing NPLEX rules here. 1 molecule of FADH2 will give us 2 molecules of ATP at the end of the day. So at the end of the electron transport chain, if we run 1 molecule of NADH through the electron transport chain, we get 3 ATP. If we run 1 molecule of FADH, we get 2 ATP. Why does NADH get more ATP than FADH? Who starts first? Yeah. NADH gets 1 more pass through a complex than FADH does. So at the end of the day, NADH is here, and then here, and then here, and then we convert our water. We take our oxygen, we convert our water. FADH only gets 2. So FADH is only generating essentially like 2 hydrogen ions. 2 NADH is 3 hydrogen ions. So that is why you get that differential. It's not like FADH. Part of it is how many electrons it can hold on to. That's why it starts later. But FADH isn't a poorer or worse molecule. It just starts later in the complexes because of the electrons that it holds. Okay. So that's our differential. NADH3, FADH2. So for 1 molecule of glucose, we go through glycolysis 1 time. We're going to have 2 NADH formed and 2 ATP formed. This is net. So this is from 1 molecule of glucose. Pyruvate, then our pyruvate conversion that happens, we have 2 molecules of pyruvate from our 1 molecule of glucose. So we're going to convert both of them to acetyl-CoA. So each of those conversions gives us 1 NADH. Since we did it twice, we get 2. Then we go through our TCA cycle twice because we have 2 acetyl-CoA for every 1 molecule of glucose. We get 6 NADH, 2 GTP, and 2 FADH from that process. And then we're going to run it through our electron transport chain. So if we just first add up stuff, we have 10 NADH, 4 ATP or GTP. They can be used interchangeably, the same, and 2 FADH. So that's what our totals are. So we just have to convert it. 10 times 3, 2 times 2, and we get 34 and 4, which equals 38 ATP from old rules or NPLEX rules. But there's only actually 36 molecules of ATP produced. Why? Using these exact rules, there's only 36, but we calculated 38. What did we do wrong? It wasn't in the math error, I promise. So there's 2 that we have to use that we didn't allocate for somehow. The pyruvate oxidation, TCA cycle, and oxidative phosphorylation all occur where in the cell? In the mitochondria. And glycolysis occurs where? Cytosol. Now, if we produce 2 ATP in the cytosol, that's fine because we can just use those. NADH, though, we can't get ATP unless if we get NADH through the electron transport chain. So NADH has to get into the mitochondrial matrix, inside the mitochondria. So in that process of having us have to get those 2 NADHs from glycolysis into the mitochondria, we have to use a shuttle. And the shuttle, there's 2 of them that you can use. One is the malate shuttle. The malate shuttle doesn't have any energy loss effect on our ATP generation. We have the malate shuttle in the kidney, the liver, and the heart, 3 specific areas where we want a lot of energy, right? The kidney, the liver, or heart are really high metabolic processes in the body. So they use this specialized shuttle that doesn't make us have to decrease any ATP. So if you utilize the malate shuttle, if you're in the kidney, liver, heart, you would truly indeed create 38 ATP for 1 molecule of glucose using NPLEX rules. 38. If you're using the malate shuttle, okay, we'll find that everywhere else in the body, and like skeletal muscle being a big place that does aerobic respiration, right? Everywhere else in the body doesn't use the malate shuttle. It's only in these 3 locations that use it. In the malate shuttle, to kind of orient you into what I'm talking about, so we were talking about glycolysis, which is represented here, and we got our 2-NADH generated. Those 2-NADH, we need to get them inside the mitochondrial matrix in here. So we use oxaloacetate and malate to do that process. So we have oxaloacetate, it can get converted to malate, which can regenerate actually our NAD. Malate can then process and move inwardly into the mitochondria freely through its own transporter, and it then gets converted into oxaloacetate, redox reaction, which then regenerates that same NADH inside of the mitochondrial matrix. So we essentially just interconvert oxaloacetate to malate, either in the cytosol or within the mitochondrial matrix to generate those same NADHs that we got from our process of glycolysis. Oxaloacetate's relationship is then we can actually convert it to aspartate, and you see that there's a relationship then to alpha ketoglutarate and glutamate. So that's kind of where your amino acid connection happens. But for purposes now, I just want you to know malate shuttle, these locations, no energy loss. So if you got 38, you would keep your 38 ATP. Okay. The difference is the glycerol 3-phosphate shuttle. This is found in brain and skeletal muscle. So 2 really important places for obviously where we use aerobic respiration. You do have a loss of 2 ATP overall through this process. Why? We're still going glucose to pyruvate. So this is our glycolysis here. Okay. In this process, through our process of glycolysis, 2 of these products, dihydroacetone, DHAP, and glycerol 3-phosphate, G3P, are produced, right? We remember the process of glycolysis. About halfway down, we split that molecule of glucose into 2 3-carbon molecules. These are these 2 3-carbon molecules. So for the NADHs produced in glycolysis, if we want to get them inside the mitochondrial matrix, we can actually do that via this interconversion. The conversion of glycerol 3-phosphate to dihydroacetone, because they can convert back and forth to each other, can actually create an FADH2 inside the mitochondria. But in doing so, we have to use an NAD on the cytosolic side. So we exchange, in the glycerol 3-phosphate shuttle, we exchange 1 NADH for 1 FADH2. Is that a 1-to-1 exchange ratio for ATP? How many ATP does NADH give us? Just 1 molecule. Yep, and what about FADH? 2. So every time we go through the glycerol 3-phosphate shuttle, we minus 1 ATP overall, because we convert 1 NADH to 1 FADH2. So if we have 2 NADHs from glycolysis in the brain or skeletal muscle, and we need to get them into the mitochondria to create energy, we're going to be down a net 2 ATP, because when it goes through the electron transport chain, it's not going to go through as an ADH anymore. It's actually going to go through as FADH2. So we'll be down 2. So 36. Yep, so here's our new picture. Well, actually, we'll do an actual now. So 36 total. So if you're thinking about NPLEX rules using the conversion factor of NADH is 3 ATP and FADH2 is 2 ATP, generation in liver, kidney, what did I say? Gut? Heart. No, yes, liver, kidney, heart. Thank you. Gives us 38. Generation in brain and skeletal muscle gives us 36. Okay, that's NPLEX rules. I do want to show you actual rules, because this is what's actually in research. And they may update NPLEX. So I want you knowing 36 and 38, and then we're going to see 30 and 32. So these are 2 numbers we're going to keep in our brain. And depending on the conversion factor they give you, if they give you one, that will give you your final number. If they don't give you one, I would use NPLEX rules of 3 and 2 on your exam, because that's what they've done traditionally. 3 ATP for 1 NADH, 2 ATP for FADH2. But in all reality, it's a little bit smaller. 1 molecule of NADH is actually 2.5. 1 molecule of FADH2 is actually 1.5. And so it ends up being 32 ATP is the true number we found, and then 30 if they use the G3P shuttle. So this is the actual number versus the NPLEX number. Yeah. Maybe based off of where it's located, yes. So they would need to give you a location, ideally, of where that tissues, where that process is occurring. So in the liver, in the skeletal muscle, in the whatever, in the brain, at the end of glycolysis, or they would tell you a place of the body, like the right shoulder, I don't know, but then you would, or there's an action, a person was running, after running, how much ATP would they generate from 1 molecule of ATP or 1 molecule of glucose? Then that's how you would know your shuttle. NPLEX is historically good at not giving you everything you need. So if they don't give you everything they need, how would I go about this? I would go with the smaller number if they don't give you everything they need. And I'd probably write a little note next to the question and be like, this is not complete. But that's just me. I wrote a lot of passive aggressive questions or notes to my question writers on my NPLEX exam. Questions about this piece, because I know this is where people get tied up. There's not going to be more than 1 or 2 questions on NPLEX, if any at all, on this ATP generation. So I would not spend a lot of time on this. Okay, great. Yes, of course. Creatine phosphate. So creatine phosphate, not a part of this process at all. Think about it, local skeletal muscle, a storage facility for ATP. Creatine phosphate is essentially a molecule that's like attached to ATP that can be released, and then ATP can be used. So it only provides like 10 to 15 seconds of ATP use. So it's less than even anaerobic glycolysis can do more. Yeah. If you didn't have, yeah. So you would probably, it would be like your ATP stores would be used first, creatine phosphate next, if it was like short sprints, then anaerobic glycolysis if you didn't have oxygen present. So that could be like a 400 meter. So if you're doing like a fast sprint for a longer duration, that's like up to a minute, then you could go into anaerobic glycolysis and create lactic acid. That's like why 400-meter runners get like that like dead leg feeling. Same with like swimmers too. And the distance running, you're typically using aerobic respiration and glycogen stores. Unless if you get into that anaerobic state, then you would start feeling, so at the end of a marathon, you sometimes see runners like, it's because they've moved into anaerobic glycolysis and they've probably ran out of stores. So their muscles tighten up. Yeah, good stuff. Gearing up to coach my track team this spring. So I'm getting ready to wow them all about biochemistry. Although they're high schoolers, so we don't go that in depth. Like, drink water. That's where we start. All right, so we've gone through the whole process, start to finish, of what we do with a molecule of glucose and how we get energy. Gluconeogenesis is just the reverse of glycolysis. There's only 3 areas I want you to worry about. There's 3 reactions that you need to know, and they are the opposite of the 3 main reactions of glycolysis, right? So step 1 of glycolysis, glucokinase or hexokinase, that is a one-way direction reaction. So for gluconeogenesis, we know we're going to need to have a different enzyme, and we do, in fact. Glucose 6-phosphatase is our separate enzyme. So that's the last step of gluconeogenesis. Then when we hit phosphofructokinase and glycolysis, that's our rate-limiting step, right? Rate-limiting enzyme. On the other end here, we have fructose bisphosphatase. So again, another unique reaction to gluconeogenesis. All these are the same because they're all reversible. So there's no change, no difference here, just the direction. We then have two reactions here that are different. Because on the glycolysis side, we have pyruvate kinase, and then we have pyruvate dehydrogenase. On this end, to get our pyruvate, 2-oxaloacetate, and then 2-phosphoenolpyruvate, we use pyruvate carboxylase, and then PEP carboxykinase. PEP is just phosphoenolpyruvate. That's what PEP stands for. This, though, is your rate-limiting enzyme, pyruvate carboxylase. So this is your rate limiter. This would be a close second, your PEP carboxylase. Either two of these would be a good answer. This one, because it uses energy, GTP. This one, because it's first, right? So these are your three reactions I'd know for gluconeogenesis. Okay. So then we have our rate-limiting steps here. Pyruvate carboxylase needs this acetyl-CoA. Phosphoenol pyruvate kinase, our PEP carboxykinase. Fructose 1,6-bisphosphatate and glucose 6-phosphatase. So again, our three, I guess, or four important reactions. And then you can kind of look here. This is from ingested glucose to when we would do glycogenolysis versus gluconeogenesis. And this is the fed state versus fasting versus starved. Yeah, does OAA still use the malate shuttle to move to cytosol for this reaction? Yes, it does. So you can see, if we go back to our malate shuttle, let me get back in here. Malate oxaloacetate. This is how oxaloacetate is able to get into the actual cytosol to be able to do this process, is using the malate shuttle. Yep. And instead of going back to malate, it would then go over here and then go and become PEP if conditions were met. Yep. Thanks, Gigi. The good thing to keep in mind, what Gigi said, it's that everything here is interconnected, right? Nothing's happening in a vacuum. All of these different reactions are occurring in the same cell, same cytoplasm, same mitochondria, same time. So the more we can kind of interconnect things, the better. That's why I encourage you, like, get a big poster board and start drawing things out over the next few weeks. Once you've drawn it out, start quizzing yourself and then draw it out again. Can be helpful. You don't have to put every single enzyme, just your rate limiters, your important ones. So a moment on regulation of metabolism. So how does the gut relate to this whole process? So there is hormones involved, right? So insulin and glucagon are two main hormones that are going to help us in this kind of carbohydrate regulation. Sorry, I have my little screen popped in place. How can I make this go away? I don't know. Maybe if I do this. Nope, that didn't work. Oh, that's fine. See if it'll... Okay, well, it's just going to be here. Okay, so insulin side here, glucagon side here. All right, so insulin is a polypeptide hormone. It's secreted by our beta cells of our pancreas. That's an important keynote. The secretion is upregulated or stimulated when we have a high level of blood glucose or hyperglycemia. And it's also, it's inhibited by when we have an elevated amount of epinephrine. So in a stressed state or a sympathetic dominant state, right? So we can connect it back to that autonomic nervous system. When we secrete insulin, we're going to see glucose uptake is going to increase. We will have an increased synthesis of glycogen, of protein, of fat. We're in a fed state. We got lots of stuff. We're not going to see glycogenolysis or gluconeogenesis because we don't need more sugar. We don't need breakdown of glycogen. We don't need ketogenesis, and we don't need lipolysis, right? We don't need to break down fats. We don't need to create ketones. And then all of these can alter gene expression. From here, everything is going to be mediated by activation of the insulin receptor, so insulin binding to its receptor. Activation of the insulin receptor activates receptor tyrosine kinase activity. Insulin binds to a receptor extracellularly, And then tyrosine kinase is actually the enzyme that's attached intracellularly to that receptor. And that's what becomes activated. And that's the second messenger system. We'll then see phosphorylation of the insulin receptor and insulin receptor, I think it's insulin receptor complex. And then we'll see a cascade of our cell signaling responses. Those phosphorylations of enzymes, which ultimately will lead to target proteins, whatever is needed in the body. We're going to upregulate some stuff. We're going to make sure we have everything needed to go through the whole process of glycolysis and aerobic respiration to utilize our energy. On the other end of the plan, if we are in a fasting state, we're going to be governed by glucagon. Glucagon is a polypeptide hormone. It's secreted by alpha cells of the pancreas, so not beta cells, alpha cells. It's stimulated when we're fasted, so when we don't have blood glucose or hypoglycemia. It's also stimulated by epinephrine. It's inhibited by insulin, so they're countering each other. When we have glucagon hormone release, we're going to see an increase of all the things that didn't happen before. Increase in glycogenesis and increase in gluconeogenesis. We're trying to get sugar, so we're trying to create glucose. We're going to see lipolysis and ketogenesis, so creation of ketone bodies and breakdown of fat. And we're going to see an uptake of amino acids because we're going to need to utilize whatever we can to create as much fuel as possible because we aren't getting fuel intake. All of that is mediated by our glucagon receptor, which is going to activate our classic G protein second messenger system. That's going to be our adenylate cyclase, protein kinases, and that leads to our downstream effect. Insulin is secreted as proinsulin, and then it's then cleaved at these arrows here into insulin. Okay. So if they ask you about proinsulin, is the inactive form of insulin, needs to be cleaved to create insulin, and then C-peptide is the component that's released. I wouldn't consider it a zymogen because it's a hormone. So I would consider it because zymogen is typically like inactive form of enzyme, and I would put insulin more in a class of hormone versus enzyme. Yeah. But proinsulin, you could kind of think of it like zymogen, and that's an inactive form, and it has to be cleaved to actually be able to bind to its receptor. Neurologically, when we are in a hypoglycemic state, we have a hypothalamic regulatory center in our brain that's going to actually stimulate our pituitary gland to secrete ACTH, which is going to act on our adrenals to secrete cortisol and secrete epinephrine. Right, we secrete epinephrine from the medulla, cortisol from the cortex, and we'll actually look at cortisol secretion here, I think, soon. Our autonomic nervous system will see an increase in norepinephrine, and then our pancreas alpha cells, we see an increase in glucagon. All these things are saying, help me, I need blood sugar, right? And our brain needs glucose, so that's why part of these processes occur. We will harvest anything that we need to make sure we can feed our brain glucose and our blood cells glucose. So we will break down helpful tissue if those organs are starving. All right. Fructose and galactose, a moment on them. Fructose metabolism. So fructose comes through into glycolysis at the DHAP glyceraldehyde 3-phosphate stage. So we can see here that fructose utilizes an ATP, get converted into fructose 1-phosphate. And that uses fructokinase, kind of like hexokinase. So we're at the equivalent of 1 ATP use. Then fructose 1-phosphate can become dihydroxyacetone and go directly into glycolysis here using aldolase B. And then glyceraldehyde, this is different than glyceraldehyde 3-phosphate. So glyceraldehyde has to take one more step where it's converted into glyceraldehyde 3-phosphate, which then can enter into our glycolysis process. And this process does take an ATP. So for 1 molecule of fructose, we are using 1 more molecule of ATP than for glucose. Do you see how that falls? We have ATP, ATP, and the glyceraldehyde has to go one more step that normally we don't have to do. That's why, and then our rate limiting step of this process is here, fructose 1-phosphate, aldolase B, because this is the committal step. Fructose 1-phosphate, once it becomes a hydroxytone, this can actually go into the glycolysis. It can undergo normal glycolysis process. So if you have a deficiency of aldolase B, you can have fructose intolerance, meaning you're unable to process fructose sugar. So our patients, if you've ever had a patient who has trouble processing fruit sugar or fructose, they could have a deficiency of aldolase B. If you have fructosuria, it could be that you have a deficiency of fructokinase, meaning that you're not even converting fructose into its first step. So you're actually urinating it or excreting it through your kidneys. So your uptake there's no uptake or no conversion taking place. All right, galactose metabolism isn't too exciting. It's pretty similar. Galactose, you use an ATP just like you do with glucose, using galactokinase to phosphorylate it. Then you use the UDP glucose. Remember, we've seen UDP glucose before. This time, though, we're actually utilizing it to take the galactose. So UDP galactose binds, it kicks off UDP glucose, and UDP glucose now becomes glucose 1-phosphate here. And then glucose 1-phosphate can be converted into glucose 6-phosphate, which moves into glycolysis. So the real rate limiting step would be this step here, step 1, and then galactose 1-phosphate, uridyl transferase would, I guess, be your secondary, but this would be your rate limiter here. If you don't have a galactokinase, you can have non-classical galactosemia. If you can't convert this glucose galactose UDP, you have classic galactosemia. That's what these little rectangles mean, meaning you cannot break down or utilize galactose. You'll have an increased amount in the blood. We did it. Metabolism done. Okay. Take 7 minutes. We'll come back at 2 with our next one. So stand up, stretch, do what you got to do. We'll go into part 3 next, which is going to be genetics and molecular biology essentials.